Hello and welcome friends. Today I am going to discuss the topic action potential and synaptic transmission. The main objectives of this lecture are to understand the specialized membrane properties of nerve cells. Second, the ability of nerve cells to produce action potentials, and to explore the basic mechanisms of synaptic transmission. Now the introduction about the nervous system. The nervous system coordinates the activities of many other organ systems. It activates muscles for movement, controls the secretion of hormones from glands, regulates the rate and depth of breathing, and involved in molting and regulating the multitude of other physiological processes. To perform these functions, the nervous system relies on neurons, which are designed for the rapid transmission of information from one cell to another by conducting electric impulses and secreting chemical neurotransmitters. The electrical impulses propagate along the length of nerve fiber processes to their terminals, where they initiate a series of events that cause the release of chemical neurotransmitters. The release of neurotransmitters occurs at sites of synaptic contact between two nerve cells. Released neurotransmitters bind with their receptors on the postsynaptic cell membrane. The activation of these receptors either excites or inhibits the postsynaptic neurons. The propagation of action potentials, the release of neurotransmitters, and the activation of receptors constitute the means whereby the nerve cells communicate and transmit information to one another and to non-neural tissues. In this lecture, we examine the specialized membrane properties of nerve cells that endow them with the ability to produce action potentials, explore the basic mechanisms of synaptic transmission. Now the special anatomical features of neurons. The shape of a nerve cell is highly specialized for the reception and transmission of information. One region of the neuron is designed to receive and process incoming information. Another is designed to conduct and transmit information to other cells. Emerging from the soma, the cell body of a neuron, are processes called the dendrites and axons. Many neurons in the central nervous system also have knob-like structures called dendritic spines, extend from the dendrites. The dendritic spines, dendrites and soma receive information from other nerve cells. The axon conducts and transmits information and may also receive information. Some axons are coated with myelin lipid structure formed by the glial cells, oligodendrocytes in the central nervous system, or Schwann cells in the peripheral nervous system. Regular intermittent gaps in the myelin sheath are called nodes of Ranvier. The speed with which an axon conducts information is directly proportional to the size of the axon and the thickness of the myelin sheath. The end of the axon terminal, contains small vesicles packed with the neurotransmitter molecules. The site of contact between a neuron and its target cell is called a synapse. Now the channels allowing ions to flow through the nerve cell membrane. Ions can flow across the nerve cell membrane through three types of ion channels. First, non-gated or the leakage. Number second, ligand-gated and the voltage-gated channels. Now first, non-gated ion channels. Non-gated ion channels are always open. They are responsible for the influx of sodium ion and efflux of potassium ion when the neuron is in its resting state. Ligand-gated ion channels directly or indirectly activated by the chemical neurotransmitters binding to the membrane receptors. When chemical transmitters bind to the receptors, the associated ion channels can either open or close to permit or block the movement of specific ions across the cell membrane. Voltage-gated ion channels are sensitive to the voltage differences across the membrane. In their initial resting state, these channels are typically closed. They open when a critical voltage level is reached. Now the electrical properties of neuronal membrane. Electrical properties of the neuronal membrane play important roles in the flow of ions through the membrane, the initiation and conduction of action potentials along the axon, and the integration of incoming information at the dendrites and the soma. These properties include membrane conductance and capacitance. Now, the resting membrane potential of nerves. The resting membrane potential of large nerve fibers when not transmitting neurosignals is about minus 90 millivolts. That is, the potential inside the fiber is 90 millivolts more negative than the potential in the extracellular fluid on the outside of the fiber. Active transport of sodium and potassium ions through the membrane sodium-potassium pump. All cell membranes of the body have a powerful sodium-potassium pump that continually transports sodium ions to the outside of the cell and the potassium ions to the inside. Further, note that this is an electrogenic pump because more positive charges are pumped to the outside than the inside. Means 3 sodium ions to the outside for each 2 potassium ions to the inside, leaving a net deficit of positive ions on the inside. This causes a negative potential inside the cell membrane. The sodium-potassium pump also causes large concentration gradients for sodium and potassium across the resting neuromembrane. An action potential depends on the presence of voltage-gated sodium and potassium channels that open when the neuronal membrane is depolarized. When the axon is depolarized slightly, some voltage-gated sodium channels open as sodium ion enter and cause more depolarization. More of these channels open. At a critical membrane potential called the threshold, incoming sodium ion exceeds outgoing potassium ions through leakage channels, and the resulting explosive opening of the remaining voltage-gated sodium channels initiate an action potential. The action potential then propagates to the axon terminal, where the associated depolarization causes the release of neurotransmitter. The initial depolarization to start this process derives from synaptic inputs, causing a ligand-gated channels to open on the dendrites and somata of most neurons. Now, the characteristics of the action potential. Depolarization of the axon hillock to threshold results in the generation and propagation of an action potential. The action potential is a transient change in the membrane potential characterized by a gradual depolarization to the threshold, a rapid rising phase, an overshoot, and a repolarization phase. The repolarization phase is followed by a brief after hyperpolarization before the membrane potential again reaches resting level. Now, the initiation of action potential. In most neurons, the axon hillock is the trigger zone that generates the action potential. The membrane of the initial segment contains a high density of voltage-gated sodium and potassium ion channels. When the membrane of the initial segment is depolarized, voltage-gated sodium channels are opened, permitting an influx of sodium ions. The influx of these positively charged ions further depolarizes the membrane, leading to the opening of other voltage-gated sodium channels. This cycle of membrane depolarization, sodium channel activation, sodium ion influx, and membrane depolarization is an example of positive feedback, a regenerative process that results in the explosive activation of many sodium ion channels when the threshold membrane potential is reached. If the depolarization of the initial segment does not reach threshold, then not enough sodium channels are activated to initiate the regenerative process. The initiation of an action potential is therefore an all or none event. It is generated completely or not at all. Now, the propagation and speed of action potential. After an action potential is generated, it propagates along the axon towards the axon terminal. It is conducted along the axon with no decrement in amplitude. The mode in which action potentials propagate and the speed with which they are conducted along an axon depends on whether the axon is myelinated. The diameter of the axon also influences the speed of the action potential conduction. Larger diameter axons have faster action potential conduction velocities than the smaller diameter axons. Now the synaptic transmission. Neurons communicate at synapses. Two types of synapses have been identified up to yet. Number one, electrical synapse, and the second is the chemical synapses. At electrical synapses, passageways known as gap junctions connect the cytoplasm of adjacent neurons and permit the bidirectional passage of ions from one cell to another. Electrical synapses are uncommon in the adult mammalian nervous system. Typically, they are found at dendritodendritic sites of contact. They are thought to synchronize the activity of neuronal populations. Gap junctions are more common in the embryonic nervous system, where they may act to aid the development of appropriate synaptic connections based on synchronous firing of neuronal populations. Now the electrical synapse. Electrical synapses have two main advantages. First, faster communication because action potentials conduct directly through gap junctions. Electrical synapses are faster than chemical synapses. Second, synchronization. Electrical synapses can synchronize, coordinate the activity of a group of neurons or muscle fibers. Now the chemical synapses. Although the plasma membranes of presynaptic and postsynaptic neurons in a chemical synapse are close, they do not touch. They are separated by the synaptic cleft, a space of 20 to 50 nanometer that is filled with interstitial fluid. Neuroimpulses can't conduct across the synaptic cleft, so an alternative, indirect form of communication occurs. In response to a neuroimpulse, the presynaptic neuron releases a neurotransmitter that diffuses through the fluid in the synaptic cleft and binds to receptors in the plasma membrane of the postsynaptic neuron. The postsynaptic neuron receives the chemical signal and in turn produces a postsynaptic potential, a type of graded potential. Thus, the presynaptic neurons converts an electrical signal into a chemical signal. The postsynaptic neuron receives the chemical signal and in turn generates an electrical signal. The time required for these processes at a chemical synapse, a synaptic delay of about 0.5 milliseconds, is the reason that chemical synapses relay on signals more slowly than the electrical synapses. A typical chemical synapse transmits a signal as follows. First, a neuroimpulse arrives at a synaptic end bulb of presynaptic axon. The depolarization phase of the neuroimpulse opens voltage-gated calcium ion channels, which are present in the membrane of synaptic end bulb. Because calcium ions are more concentrated in the extracellular fluid, calcium ions flows inwards through the open channels. Third, an increase in the concentration of calcium ions inside the presynaptic neuron serves as a signal that triggers exocytosis of the synaptic vesicle. As vesicle membranes merge with the plasma membrane, neurotransmitter molecules within the vesicles are released into the synaptic cleft. Each synaptic vesicle contains several thousands of molecules of neurotransmitter. Fourth, the neurotransmitter molecules diffuse across the synaptic cleft and bind to neurotransmitter receptors in the postsynaptic neurons plasma membrane. Fifth, binding of neurotransmitter molecules to their receptors on ligand-gated channels open the channels and allows particular ions to flow across the membrane. Sixth, as ions flow through the opened channels, the voltage across the membrane channel changes. This change in membrane voltage is a postsynaptic potential. Seventh, when a depolarizing postsynaptic potential reaches threshold, it triggers an action potential in the axon of postsynaptic neuron. At most chemical synapses, only one-way information transfers can occur from presynaptic neuron to a postsynaptic neuron or an effector, such as a muscle fiber or a gland cell. Only synaptic end bulbs of presynaptic neurons can release neurotransmitters, and only the postsynaptic neuron membrane has the receptor proteins that can recognize and bind that neurotransmitter. As a result, action potentials move in one direction. Now, the neurochemical transmission. Neurons communicate with other cells by the release of chemical neurotransmitters, which act transiently on postsynaptic receptors and then must be removed from the synaptic cleft. Transmitter is stored in synaptic vesicles and released on neurostimulation by the process of exocytosis, following the opening of voltage-gated calcium ion channels in the neuroterminal. Once released, the neurotransmitter binds to and stimulates its receptors briefly before being rapidly removed from the synapse, thereby allowing the transmission of a new neuronal message. The most common mode of removal of the neurotransmitter following release is called as a high affinity reuptake by the presynaptic terminal. This is a carrier-mediated sodium-dependent secondary active transport that uses energy from the sodium-potassium ATPase pump. Other removal mechanisms include enzymatic degradation into a non-active metabolite in the synapse or diffusion away from the synapse into the extracellular space. There are several classes of neurotransmitters. The first neurotransmitter is acetylcholine. Neurons that use acetylcholine as their neurotransmitter are known as cholinergic neurons. Acetylcholine is synthesized in the cholinergic neurons from choline and acetate under the influence of the enzyme choline acetyl transferase or choline acetylase. This enzyme is localized in the cytoplasm of cholinergic neurons, especially in the vicinity of storage vesicles, and it is an identifying marker of the cholinergic neuron. Now, the next, catecholamines. The catecholamines are so named because they consist of catechol moiety and an ethylamine side chain. The catecholamines, such as dopamine, norepinephrine and epinephrine, share a common pathway for biosynthesis. Epinephrine secreting cells include a small population of central nervous system neurons as well as the hormonal cells of the adrenal medulla. Chromaffin cells which secrete epinephrine during the flight or fight responses. Most of the catecholamines released into the synapse is rapidly removed by uptake into the presynaptic neuron. Once inside the presynaptic neuron, the neurotransmitter enters the synaptic vesicles and is made available for recycling. Serotonin. Serotonin is a transmitter in serotonergic neurons. Chemical transmission in these neurons is similar in several ways to that described for catechol-aminergic neurons. Gamma-aminobutyric acid and glycine. The inhibitory amino acid transmitters, gamma-aminobutyric acid or GABA and glycine, bind to their respective receptors, causing hyperpolarization of the postsynaptic membrane. GABAergic neurons represent the major inhibitory neurons of the central nervous system, where glycinergic neurons are found in limited membrane restricted only to the spinal cord and brain stem. Now, the neuropeptides. Neurally active peptides are stored in synaptic vesicles and undergo exocytic release in common with other neurotransmitters. Many times, vesicles containing neuropeptides are co-localized with vesicles containing another neurotransmitters in the same neuron, and both can be shown to be released during neurostimulation. With this, we conclude today's lecture, Action Potential and Synaptic Transmission.